So an important fact that I want to point out, if we haven't quite made this connection yet, is yes, I put cell membrane off to the side, but please keep in mind that this same type of membrane with the phospholipid bilayer, with the proteins that are embedded in that lipid bilayer, with all the other pieces and parts attached, that is going to be really the same general structure that we see in the nuclear membrane, in the endoplasmic reticulum, in all of these membrane-bound organelles. They're all going to have membrane, and they're all going to basically have the same structure. There's differences relating to exactly what the function of the organelle is, but in general, they all are phospholipid bilayer, proteins, etc. Okay, and so for that reason, the transport that we're about to talk about, yeah, that's right, is also the same kind of concept. It's not just happening into and out of the cells, but it's happening in each membrane-bound organelle as well. So when we talk membrane transport, there's really two main types, what is called passive and what is called active. Today, we're going to focus on the passive. I'm going to mention active transport only in very general terms because I don't want it to be too overwhelming. So before we go any further and actually talk about stuff moving across the membrane, let's make sure that we do understand how stuff moves in general and talk about this process of diffusion. I feel like you guys were getting it pretty well, so I don't want to beat a dead horse, as it were. But remember that when we're talking about diffusion, we're just talking about molecules or some type of particle that is spreading out, going from an area of high to low concentration. But it's the net movement, the overall movement that we're talking about when we say high to low concentration, because each individual particle is going at complete random directions. Each particle is just kind of bouncing off whatever's near it. I bring this back time and time again to just picture a whole bunch of second graders that are brought into a giant gym. And at first, they're going to be kind of all clustered by the door. But pretty soon, they're running around and bouncing off of each other completely at random because that's what they do. And so they start to fill up the entire gymnasium pretty much equally. Now, if you lower their energy level, bring a bunch of second graders in first thing in the morning when they're super tired on the first day of school, and they move much slower. Pop them up on Halloween candy, way more energy. They're much faster, and that process is going to happen faster where they fill up the entire area. Particles are acting the same way. Low energy, they're slow. High energy, they're fast, and diffusion happens faster. And that energy is temperature. That's thermal energy. Okay. Now, surface area to volume ratios are going to be incredibly important as they relate to how a cell is able to function. So think of it like this. When I say surface area, I really mean what's covering the cell. In this case, that would be the cell membrane. So the cell membrane is being represented by this measurement of surface area, and that's where materials can enter and exit. That's where transport happens. The rest of the cell, everything inside of that, is what's causing transport to be necessary. So nutrients are constantly being used up. So we need to constantly draw in more nutrients so that the volume of the cell, the cytoplasm, has what it needs. This volume, the cytoplasm, is what is producing the waste products that then have to get out. So if you're a cell and you're this big, and then you grow to this big, your surface area has gotten bigger, but your volume has gotten bigger faster because surface area is going to grow, as you saw even just with the activity this morning. Surface area is going to grow in a square root or in a square. That's what I mean. The formula has a square in it. Whereas the volume is going to increase by a cube, which means that that volume is growing exponentially faster. So if I'm comparing surface area to volume, even though surface area is getting bigger, volume's getting bigger faster, and we're going to know that the ratio decreases. Underline, sparkly, highlight, whatever you want to do. Important idea here. As the surface area to volume decreases, efficiency drops. All right, so I know you guys haven't had a chance to really go shopping in a while, not the way that it used to be, right? But here's what I want you to picture. West Manchester Mall, you know how they now have all the outdoor shops, right? How easy or difficult is it to get in and get out to a small little store that is in those shops? Pretty easy, right? You get in, find what you need, you get out. If that store is really, really crowded and the fire alarm goes off, how long do you think it would take for the store to be evacuated? Can everyone get out pretty quickly? Okay. Now how about imagine you're in the middle of like a mega Walmart. Much bigger. Yeah, there's more doors. Yeah, there's a bigger outside perimeter. But how easy is it to get in and out? Gotta park, gotta walk 87,000 miles. If there's a fire alarm, you can bet that you're right in the middle, because that's always the luck. And it's gonna take forever to get out. It's far less efficient. In that very same way, cells that are very, very small are much, much more efficient. Stuff can get in and out really easily. If you're really, really big, not so good. Yes, so really what it's coming down to is as that surface area to volume ratio drops, efficiency drops. You become a less efficient cell. So what can you do if you're a cell and you're kind of big? What are your options? How can you increase your surface area to volume ratio? What do you think, Sarah? Hey, you can fold. It's why when we look at a lot of cells, especially in the digestive tract, they have these huge like folds that come in and out of them, those microvilli that are going to really increase how much material can move across the cells. What else could you do? Either increase your surface area, split, or decrease your volume. So that's what's going to keep cells really limited in size. So even though you're bigger than a mouse, I hope, unless it's a rodent of unusual size. Those are terrifying. But for the most part, you guys are bigger than mice. It's not because your cells are bigger. Your cells are the same size. You just have more of them. Okay. How are we doing with all that? Are we managing it? So let's get into the passive transport. This one hopefully will be quick and easy, and I'll give you guys some time to work on finishing up the pivot from yesterday. So when it comes to passive transport, there are 3 things that you must know, okay? 3 things that you must know as it comes to passive transport. Number 1, particles are always going to be moving from areas of high to low concentration. They do that anyway, which means that energy is not required to be added by the cell. And this is just going to keep on keeping on until you reach a stage of dynamic equilibrium, at which point there is no longer net movement.
Miss Unger,
Yes, indeed.
during dynamic equilibrium, you said there's no net movement. So do the particles just stop, or are they?
No, the particles absolutely continue to go through, but they basically equal out because they're going just as much in one direction as they are in the other direction. So at first, when there's like this net movement, if there's a really high concentration difference, for example, what we're going to see is overall, there's way more particles going where there's a higher concentration to where there's a lower. But then as you reach that point of like equilibrium, then the molecules are still moving, but now they kind of cancel each other out because just as many go in one direction as the other. Great question. And I think that that's one that a lot of people, especially in regular bio, like in first year bio, miss that idea. But particles are definitely still moving. And so we can look at simple diffusion things, as we said the beginning of the week, things that are small and nonpolar can go right on through the phospholipid bilayer. So this is going to be the most rapid form of transport. For facilitated diffusion, anything that is large or polar cannot simply go directly through the membranes. And so we're going to need a transport protein. Let me move myself out of the way here. So transport protein does exactly that. It's allowing for the transport through the phospholipid bilayer or through the membrane by bypassing the phospholipid bilayer.
Miss Unger?
Yeah, go ahead.
So is that what you were talking about yesterday when you said that it was like a vesicle? Because I think you had it as one of the things branching off of that.
I would have to go back and remember what I said for that to know.
Well, it was just like you talked about like lysosomes, peroximer,
okay. And
yeah,
so those are going to be those are just different vesicles that have some kind of enzyme in them, but not necessarily because of this. This would be a little bit of a different
okay
idea. Okay. So before we even talk about the different type of proteins that it could be, I'm just going to generally call them transport proteins most of the time, other than knowing that channel proteins allow for the passage faster. They really do the same things. They're just letting particles move from one side to the other. And for this one, because I apparently like to shop way too much, we're going back to a shopping mall analogy. Only this time we're shopping with ghosts. Because obviously. Now, if you park on the side of a building, bad news. You cannot move directly through the wall to get inside. Ghosts can, but you are too big and polar and exist. So in the very same way that large or polar molecules cannot get through the phospholipid bilayer, we cannot get directly through walls. We need to go through doors. And in general, we're just going to call doors transport proteins. And there can be doors that stay open or those little rotating doors that slowly let you through. Channel proteins are like a big open door that you can just go on through. Carrier proteins are like the spinning doors. It's a little slower. There's a lot more movement. Gets the same job done. The only difference here is that that door is very, very specific to who or what can get through. If you're a channel protein, one specific particle type can get through, and that is it. If it is a sodium channel, sodium's the only thing getting through. If it's a potassium channel, that's it, potassium only.
Miss Unger?
Yeah.
Can it still go through if like sodium and like it's bonded to other things?
No, good question. So if it is even a really similar molecule, it can't go through. So if it's sodium, then only the sodium ion can get through. And if it is ionically bonded to chlorine, then that thing can no longer get through because it's now joined to something different. Actually, this is the same way a lot of drugs work. Your body can only absorb certain forms of different drugs. So when they make ibuprofen, for example, ibuprofen exists in one form that it can get into your cells, but the enantiomer, the mirror image of it, cannot get through the transport proteins, so they don't work. So that's why you can take things that are chemically very similar to something else. One will work, one won't, or one will work differently. And as we'll see later, sometimes these channels or sometimes these carriers can be blocked, but that's a tomorrow story, a metaphorical tomorrow, not an actual tomorrow. Okay. All right. Preview time. On Monday, you're going to start looking at something called osmosis. You'll take notes on it over the weekend. I'm going to put it out there now because there are some concepts that your book does a iffy job explaining and straight up don't explain. And I want to at least introduce you to some of these ideas. We are going to do a lot of calculations next week, so please bring your math brains. If you don't have one of those, see if you can borrow one. I don't know how that would work. It sounds like manslaughter, so maybe don't do that. Moving on. So, osmosis. It is a type of facilitated diffusion, but so far everything that we've been talking about has to do with the solute. But water is the solvent. And so when we look at osmosis, it is the facilitated diffusion specifically of water through aquaporins. Okay, so here's that idea that I said is not introduced in your text. And I will go over it a lot next week because it is not easy. So water potential, this term right here, water potential. Water potential says, or is basically a measurement of how likely it is water is going to move. And so water is always going to move from a side with higher water potential to lower. You don't know what that means yet, but that's okay. And water potential is going to depend on the tonicity and the pressure of a solution. So water is always going to go from where you have a higher water potential to a lower one, and that's going to be dependent on both really what's in the water and is the water being pushed, or is something pushing? Okay. So again, this is going to be in your notes for Monday. Do more with trying to make sure you're thinking about this than writing it down, because you will have it written down again and it's being recorded, okay? So when we talk about tonicity, we're talking about a comparison term. We're comparing two sides of the membrane and saying, where are the solutes? So let me give you an example. A hypertonic environment, hyper means higher than. So the hypertonic side is the side with a higher tonicity. And what that really just means is that there's a greater solute concentration and a lower water concentration. So hypertonic is higher tonicity, higher solute, but because of that higher solute concentration, you have less relative solvent. Okay. And water moves from where there's more of it to where there's less, just like everything else. Everything moves from where there's more to less. So if water is moving from where there's more to less, if you were to put a cell into a hypertonic environment, then the water inside of that cell is going to move from where there's more water inside of the cell, and it's going to exit because there's less water outside of the cell. So what we see with cells that are placed in a hypertonic solution is that for animal cells, they shrivel up. For plant cells, they shrivel up, and that shriveling also pulls them away from the cell walls, causing something called plasmolysis. You'll read about it and be like, oh yeah, we done saw that on Friday. Good stuff. Okay. The opposite of a hypertonic environment, keep in mind, these are comparison terms. You cannot just have a hypertonic environment on its own. It has to be compared to something, the other side of the membrane. So a hypotonic environment would mean that the inside of the cell is hypertonic because it's a comparison. A hypotonic, like hypothermia means low temperature. Hypoactivity means low activity. Hypotonic, low tonicity. So less solute concentration and a higher concentration of water. If you take a cell, that cell is hypertonic to pure water, always. Pause my recording. Never pump Granny full of pure water in an IV. Simple instructions for life. Also, this is important. This is why you should pee. If your body does not get rid of excess fluid in a timely manner, your blood can start to become really, really hypotonic. And what's going to happen if your blood, the liquid portion of your blood becomes hypotonic? Whoosh, water rushes in, cells go pop, but I can't do that anymore because of the mask. Thanks, COVID. And that's really sad. So good story. However, as we've already learned this year, you guys are not plants, but if you were, this hypotonic environment would be prime for you. Because remember I said that pressure has something to do with water potential? Well, as it turns out, that the cell wall of this plant cell is going to push back into the, like in towards the cell and prevent too much water from coming in. And that's going to keep the plants not only full of water, but help them stand upright, rigid, or what is called turgid, because all of that excess water is really causing them to stack up nice and tall, and that's good if you're a plant, which you're not. Okay.
Unger.
Yes.
Is this water in the solute like out? Is this outside the individual cell or like inside?
Okay, so it's always that comparison. So if what we're saying is that the water concentration is higher on the inside of the cell than in its environment. If the water, what did I just say? If the water concentration is higher on the inside, that means that the solute concentration would be lower and that the solute concentration on the outside is higher. So we kind of use them as comparative terms. But when I say something like a hypertonic environment, then what I'm saying is that the environment the cell is in is hypertonic compared to a cell itself that is hypotonic. Did that make any clarification happen?
Yeah.
Okay. All right. Lastly, does anyone wear contact lenses? Okay, if you ever read the side of your contact lens solutions, they say isotonic solution. So as it turns out, the perfect thing to be putting around our cells is something called isotonic, the same tonicity. We want equal solute concentrations inside and outside. Ooh, the picture did not say that I needed. This is just the wrong picture. Pretend it doesn't exist, I'll delete it. In an isotonic solution, our cells are happy.
Mrs. Unger?
Yep.
Is this like the normal environment a cell is in?
For our bodies, yes. For animals, yes. For plants, no. Plants actually prefer to be in a more hypotonic. So the soils tend to have a lot of water, and that's going to seep in constantly into the plant, and that's what it prefers.
Miss Unger.
Yeah.
So if the cells in our body were ever to become hypotonic or hypertonic, we would become sick then.
Correct. Yes. Well, if your blood, the liquid portion of your blood and all the rest of your body were to become too hypertonic or hypotonic, the first thing your body would do is be like, ooh, either I'm really thirsty and need to drink more, or ooh, send that to the kidneys, time to go pee. So your body regulates it pretty well to make sure that that environment doesn't ensue. But if it were to go unchecked, that's not good. That would be a disease state. Okay. So kind of speaking of all of that, let's kind of contextualize this here. Simple diffusion, right across the phospholipid bilayer. Facilitated diffusion, stuff has to move through some kind of transport protein. Aquaporins being one of those will allow for osmosis to occur. You guys looked at the stem gizmo. And an important idea with that, and really all of passive transport, is that now we're faced with having to try to maintain homeostasis. So please remember that your bodies and every cell within your body is kept at a constant difference than its environment. So we said that with simple or with passive transport, that it's always going to continue until equilibrium is reached. So what do we do to prevent equilibrium from being reached? How do we stop our bodies from being at equilibrium with the environment? You're doing it right now. I see you all. How do I stop the cells of my body from just becoming at equilibrium with everything around it? What am I doing? Ever so awkwardly. Hey, every time I pull in air, what am I doing? I'm pulling in more oxygen, and I am continuing to keep a concentration gradient around my cells so that my cells are not ever at equilibrium with their environment, because if they are, well, then no diffusion can really happen. There's no net movement of particles. I need oxygen constantly coming in. If I stop breathing, then eventually all of my body gets to the same amount of carbon dioxide, of oxygen, of sugar, of all of it. And so by having these different mechanisms to constantly bring in new materials and get rid of old materials, that's ensuring that we don't ever just reach equilibrium with our bodies or with the environment. Okay. Okay. So let me do this thing.